Hi, my name is Thomas, and on the Darwin200 UK Voyage I have been studying plankton and their distribution around the British Isles. Planktonic organisms are those that drift with the current. They can be tiny microscopic organisms or as large as jellyfish. Plankton is very important because it forms the basis of the marine food web, so everything from small fish to large whales rely in one way or another on plankton. And it's split into two main groups, so phytoplankton, which are photosynthetic organisms, and zooplankton, which are kind of animal-like organisms. We have sampled mainly the British northwest coast, so from Liverpool up into more remote areas of Scotland. We have trawled the top 4 metres of the water column with a plankton net, so collecting all the microscopic organisms present in the water column, and then we have analysed them under a light microscope. Some of the species that we observed most regularly in our sampling include diatoms. These are small unicellular algae that often link together to form chains, and we also saw many dinoflagellates, so these have flagella which enable them to move through the water. In terms of zooplankton, we frequently observed copepods. These are small crustaceans which are the most abundant group of zooplankton. In addition, we saw many crustaceans and crustacean larvae, so things like crabs and their larvae, shrimps, and especially krill, which is really important in the marine ecosystem, as well as organisms such as mollusc larvae. Some of my favourite finds when sampling on this voyage have been Tomopterus, which is an annelid worm which is actually visible with the naked eye. I found them particularly interesting. Some industrial areas, for example in the River Mersey in Liverpool, had a large amount of suspended sediment particles, and this would have prevented phytoplankton from being able to photosynthesize, and so as a result there was also a lack of zooplankton in the samples. We scored each of the samples we took bad, moderate and good. So bad being very low numbers and diversity of plankton, and good being close to the natural environment. Overall, we found that there was a much higher diversity of plankton present in samples taken from more rural areas, especially those in the remote islands of Scotland. Some places came in between. Near the harbours of Tobermory and Stornoway, we found that these scored moderate on our scoring system. It was really nice to see that the results we obtained seemed to follow Water Framework Directive patterns for good, moderate and bad water quality that we found within our areas of study. Unfortunately, we were only able to collect samples from a limited number of locations due to time and weather constraints, and so it would have been nice to obtain a more thorough perspective of plankton in the British Isles, but I do think that this method that we used could definitely be rolled out across a global voyage to study plankton worldwide.